Hey, this is Taylor Corley,
And this is Hunter Appleton.
and this is our presentation on increasing diving capacity for the Lontra canadensis North American river otter. So why do we choose otters? We chose otters because they're a semi-aquatic mammal and they can make use of diving or improved diving because they rely on diving for their food. And so naturally this diving capacity will vary based off of where they're from. Like you have otters that live in more mountainous ranges, they'll have higher altitudes but less available oxygen. And in one problem that a population like this faces is at Yellowstone National Park, and this is where there's an hypoxic environment because of higher altitudes, but they have challenges with the current scarcity of food for a particular fish they eat. And so our goal is to enhance their diving capacity so they're able to uh, dive for longer periods to try to retrieve that food during the scarce times.
While myoglobin is found in the red muscles of animals, and it's used mainly for storing oxygen and facilitating it to muscles in hypoxic times. So when the oxygen is more deprived, say in diving or something like that, they're going to need more storage of oxygen. So if we can enhance the storage function of the myoglobin and we can increase the amount of myoglobin by reducing aggregation, then it could help them to store more oxygen and dive for longer and help them to retrieve more food, especially in those scarce times. Myoglobin is a globular structure. It has 153 amino acids and 8 alpha helices. In the center is the heme group, and the heme group is consisted of a porphyrin ring with an iron center. The iron center is actually what binds the oxygen, and it undergoes two conformations to bind and release oxygen. So in the R state, it binds to oxygen, and in the T state is when it's not bound to oxygen.
So according to the article Evolution of Mammalian Diving Capacity Traced by Myoglobin and Net Surface Charge, there's a few changes that can increase diving capacity. And one is increasing the myoglobin's surface net charge, which could potentially prevent myoglobin molecules from aggregating together as there can be more repulsion, and so there can be a higher concentration of them within a certain area. And another way of doing this is to replace neutral amino acids or negative amino acids with positive amino acids such as lysine or arginine. And you can also add internal histidine residues as they can be partially positive. They're not really beneficial for adding to the surface because they can dissociate, and you can also replace these two positions are key for dimer formation. So if you replace these two, then they're less likely to form dimers. So the whole goal is to increase the concentration of myoglobin within a certain area so it can take up more oxygen. So this is based off that same article. This is just a graph that's based off their results, and there is a clear linear result between the maximum of myoglobin concentration and then the total diving time. So for specific changes to myoglobin, we compared the amino acid chain of myoglobin to a whale, which obviously has really long diving times, as they're able to dive for hours at a time. And so these are the key differences that we found. There was more differences, but these were the key that we think we could hone in on that could increase the diving time. So we have this conversion at position 12, just to, again, position 12 and 122 are the ones that reduce dimer formation. So we're changing these two into more positively charged amino acids. And then this change at position 57 and 113 is both aimed to move this negative charge into a neutral charge and then move this neutral charge to positive. So they'll both increase the net surface charge. And then that's the same thing with this last one, is just moving it to a lysine just so that you can increase your surface charge.
So for the methods behind this, for our sequence analysis, we compared the myoglobin gene structure from a whale and an otter using the MegaX bioinformatics tools, and we identified certain amino acids in the whale's genome that helps them to dive at longer times, and how we could apply those to the otter's genome. So the structural insights provided by this, we could target those said substitutions in surface exposed residues and the heme binding regions. That way we could increase oxygen affinity but also prevent aggregation. So if we increase the net positive charge, that would prevent the aggregation. That way we could have higher concentrations of myoglobin. And then some proposed modifications as well for key positions such as the N12, we could swap that to K. And then for D122, we could substitute that to E. That would stabilize dimer formation and optimize like oxygen diffusion, so that way we could appropriately diffuse this oxygen to these depleted tissues. Our contributions, Hunter, I contributed to the formatting, data collection and analysis, some citations. I did a lot of the image sourcing, and I was the brain behind the methodology.
This is Taylor, and I contributed to also with some formatting and data collection. I did the proposed changes and sourced the background information. Thank you, and these are our references.